Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Rabbi shrah li sadri wa yassir li amri wahlul 'uqdatam min lisani yafqahu qawli. Rabbi zidni ilma. Okay, in today's lecture, we are going to talk about the next thing, which was the part of cell cycle, which is a type of cell division actually called as meiosis. It is second type of cell division which is responsible for the formation of cells having half the number of chromosomes as compared to the original parental cell. Before meiosis, we have talked in detail about the mitosis, in which cells were being formed having identical number of chromosomes as their parental or the original cells. But now we are talking about those cells which are formed having half the chromosomal number. What is the significance of formation of the cells having half the number of chromosomes is clearly significant, or is clearly understandable when we talk about the sexual reproduction or the fertilization. So meiosis is the type of cell division which is required for the formation of gametes, which are the cells which are responsible for the sexual reproduction, or you can say fertilization. Here we can see the form of cell division by which gametes with half the number of chromosomes are formed is called as meiosis. So originally the parental cell from which the haploid cells are going to be produced, or the gametes are going to be produced, is called as diploid cell. 2n means normal chromosomal number. 2n is the number which shows the doubled up number of a chromosome as compared to a haploid cell. So diploid cell is used for a normal somatic cell having complete number of chromosomes, while the haploid cell, which is denoted by n, is actually specified for a type of cell which is having half the chromosomal number as compared to a normal original parental cell. So these two terms are going to be repeatedly used in case of meiosis specifically, in which from one single diploid cell, 4 haploid cells are formed after the two stages of the meiotic division, that is meiosis 1 and meiosis 2, or the first meiotic division and the second meiotic division. So meiosis is a sexual reproduction process in which sexually active gametes are going to be formed. Two divisions, as I have already mentioned, that is going to be consisting of 2 stages, 2 steps, or 2 divisions, that is first meiotic division and the second meiotic division. Here we have just talked about the fertilization. What is the term? That is the fusion of sperm and an egg to form a zygote. So sperm is actually a male gamete and the egg is actually the female gamete. So during fertilization process, normal reproduction process, a sperm that is from the male side, it fertilizes an egg which is from the female side to make a zygote, or the first complete individual cell which grows into a complete individual organism. And this is not specific only in case of humans, rather it is same for the plants and the animals or any eukaryotic cell you can find in the world. A zygote is a fertilized egg which has been formed by the combination of 2 gametes. And as we have just discussed, that one sperm, which is actually one gamete, is having chromosomal number, for example 23. If I'm talking about humans, so sperm is having 23 number of chromosomes and egg is again having 23 number of chromosomes, which is actually half the number. So both of these are going to be combined and get together to form 46, which is a diploid, which is a normal cell in case of human race. So every single species is actually recognized by the chromosomal number it maintains, and from the first human on earth till the last human on earth, the chromosomal number is going to be constant. That is the exact number, which is 46, and that remains constant because of this meiotic division, as the gametes do not have 2n chromosomal number. Otherwise, for every single generation that is coming, the chromosomal number might have been doubled up and on and on and on. So the chromosomal number might have been gone into a huge sum. So this is something which is responsible for the maintenance of the chromosomal number in a specific species, whether it's plants, it's animals, or humans, or any eukaryotic cell which is forming new individuals after sexual reproduction. Here we specifically talk about the meiosis in which the sex cells divide to produce gametes, and gametes have half the number of chromosomes, and it occurs only in gonads, that is the testes in case of males and ovaries in case of females. The male process of formation of male's gametes is called as spermatogenesis, and in case of female, the formation of eggs or the female gametes is called as oogenesis. Meiosis is similar to mitosis with some chromosomal differences, as we have talked about that yes, the chromosomal number of the resultant cells are going to be half. Here we have spermatogenesis. So a human sex cell which is diploid in number is first going through first meiotic division, in which the chromosomal number is going to be just halved, that 23, and finally each cell is going to maintain the same chromosomal number as compared to the middle one, and that is the meiosis II. And here you can see meiosis II is almost similar to mitosis, in which the chromosomal number is going to be conserved in case of second meiotic division, but in case of first meiotic division, the chromosomal number is going to be halved. That's why the first meiotic division, or meiosis first or meiosis I, is also called as reductional division or reduction division, in which the chromosomal number reduce to half. In case of interphase 1 that we have already talked about, in case of mitosis, there's a preparatory phase in which cell is actually going to be preparing itself for the purpose of cell division. Same like that, in meiosis, cell is also going to be preparing itself for the next cell division. So similar to mitosis, interphase is going to be occurred in case of meiosis as well. Chromosomes are going to be replicated, which is actually the S phase. Each duplicated chromosome consists of two identical sister chromatids attached at their centromere, same like we had previously. Centrioles pairs also replicate, so there are two centrioles each on the sides. Interphase 1, first of all, the nucleus and nucleolus are visible right here. The chromatin is there, cell membrane is there, nucleolus is there, nuclear membrane is there. So everything is there before the prophase begins. Now, first meiosis is also consisting of four phases, that is going to be specifically prophase 1, metaphase 1, anaphase 1, and telophase 1. Prophase 1 in this case of meiosis is going to be the longest and the most complex phase, in which the major or the most important events occur, which are responsible for the specific significance of the meiosis. So it is consisting of 90% of the whole cell division process, in which some certain things are just like mitosis. For example, chromosomes are just going to be condensed, or the chromatin is going to be condensed into chromosomes. But the most important thing that is going to occur here is the most important event called as synapsis. Now, what is synapsis? Before going into the detail of synapsis, we have a term called as homologous chromosomes. Homologous chromosomes are such chromosomes which are identical in their shape, in their structure, and the position of their centromere. So we have got multiple chromosomes in our DNA, and the chromosomes which are actually identical in their shape and structure, which are identical at their central position or the position of centromere, are called as homologous chromosomes. So in the step of synapsis, the similar chromosomes or the homologous chromosomes get together to make pairs, and these pairs are called as tetrads, because in pairs there are total 4 chromatids. There are 4 chromosomal arms. That's why it is called as tetrad. Or tetrad is 2 chromosomes or 4 chromatids, sister and non-sister chromatids. That is the differentiation we have already talked about in mitosis. Here we can see these are sister chromatids, and the completely red one and the completely blue one are actually red and blue just to differentiate that these are 2 different chromosomes, but according to the position of their centromere, according to their size, according to their shape, they are perfectly similar. So that's why these chromosomes are just going to be paired up, and this pair is actually having 4 chromatids, 1, 2, 3, 4 chromatids. That's why it is called as tetrad. And it is also called as bivalent because individually these are 2 chromosomes. Bi means 2. So bivalent or tetrad. So these homologous chromosomes are just paired up and this process is called as synapses. After the pairing of chromosomes, which are homologous in their nature, one is from mother side, other one is from paternal side. They are similar in shape and size and also called as tetrads, have some inherited traits. How? Because they have certain genes which are controlling certain activities of the body, and each locus is actually the position of the gene on the chromosome. So every single chromosome has a specific position of a gene. For example, at position A of chromosome, if I just go back to the previous slide, for example, I'm talking about this one purple thing. For example, it is having position A for some eye color, and the next chromosome maybe is having on the same position A that is having the gene for insulin. So the position of genes is not specifically same in all the chromosomes. For example, in this room, you are sitting on the front line chair, and the same person if he gets a chance to go to the next room. For example, if he is just going to fit into this chair which is on the third row. So that is the position which is not specific. So similarly, these are two different chromosomes because this is one person, but that one person has got these chromosomes from his mother and father. So he has got two different chromosomes from his father line and mother line, and these chromosomes are actually having genes which is having different position on both the chromosomes. As humans have total 46 chromosomes, that means they have 23 pairs of homologous chromosomes. So 22 pairs are called as autosomes, and one pair is called as sex chromosome, which is actually defining the person's gender, whether it's going to be male or female. Now in that case, the homologous chromosomes which are actually paired up, now first thing on the first chromosome is eye color located, and the second is having eye color located, but the eye colors are, for example, different. And similarly, from paternal side, hair color is, for example, brown, and from maternal side, hair color is going to be black. So that are the gene which are similar in their position but having different properties. Now in case of chromosomes, so this is something which is actually responsible for the what I was telling, that these are the chromosomes which are identical, which are 22, while the 23rd chromosome is actually different, which is showing the XY chromosome. X is the female and Y is the male chromosome, and that is what defining the gender of the produced cell or the zygote. So that the Y chromosome decides whether the person is going to be a boy or it is going to be a girl. So continuing with the point of the synapses that we were talking about, the two homologous chromosomes actually have got paired up, and after pairing up, they are going to exchange their parts. What is meant by exchange their parts? They are going to cross over, and crossing over may occur between non-sister chromatids at the chiasmata. Chiasmata is the position or the central point at which the two homologous chromosomes have been paired up. So the crossing over segments of non-sister chromatids break and reattach to the other chromatid, and the chiasma are the sites of crossing over. For example, there were two non-sister chromatids which were actually paired up during synapsis, and this pairing is the synapsis is between the two chromosomes which are same in their structure and their shape but are from two different lines, one from mother and one from the father. And you can see here in the central point that the two arms of the non-sister chromatids are just going to be exchanged. The purple one is going to give its part to the red one, and the red one is going to give its part to the purple one. And finally, we have got a variety, or we have got chromosomes which are actually having shuffle genes. So now one single chromosome which was actually before that position, the single chromosome which was having only one type of properties from either mother or from the father, but now finally after meiosis we have got a chromosome which is actually having both the properties mixed together. And this is the reason that whenever the chromosome is just going to be participating during fertilization, it is actually having both the varieties or both the genes from their father and mother. So that's why the offspring that is going to be developed or that is going to be produced may have properties of their grandparents from mother side or from father side. So this is the basic backbone of the variation that is produced after the sexual reproduction or sexual fertilization. That is the backbone of the variety that is being produced. And this is the same reason that even if the same